Good morning, everyone. Welcome to our channel. So for today's video, Joanson Powder, our group, will discuss to you Chapter 11, which is the learning, memory, and amnesia. What is learning and memory? Learning and memory are two ways of thinking about the same thing. So basically, learning and memory are closely related concepts. Learning is the acquisition of skills and knowledge. Example is student who understands and remember what have been taught to us. And what understanding will you get for today's discussion is learning as well. While memory, on the other hand, is the expression of what have you acquired. Another difference is learning is acquiring knowledge and skills slowly. It is basically deals with the experience changes in the brain, while memory is acquired instantly. This is how data are stored and subsequently reactivated. Without learning and memory, we would experience lifeless life, as if we are waking from a lifelong sleep since people around you will be a stranger. And every time you wake up, things are new to you. So learning and memory really is important in human life, since it basically revolves around our daily lives. So if we don't have learning and memory, then everything will be strange to us. So here are the chapter overview and learning objectives, which you can see it in the book. You can just read it. But basically, this chapter will focus on the roles played by the various brain structures of processes of learning and memory. And we will also encounter in this chapter the study of neuropsychological patients with brain damage that produces amnesia. Before we go through formal assessment of HM's interrogate amnesia, discovery of unconscious memory, let us first know who is HM. Henry Molaison, also known as HM in psychology field, is a man who in 1956, at the age of 27, has medial portion of his temporal lobes removed for the treatment of severe case of epilepsy. His case was the instrument in the achievement of the current understanding of neural brain memory. HM went under the bilateral medium temporal lobectomy. So as you can see in this picture, this is what the actual normal brain looked like, and this is the brain of Henry Molaison. This is the procedure that he went under, which is the bilateral medial temporal lobectomy. So it is the removal of the medial portion of both temporal lobes, including most of the hippocampus, which is the major role in learning and memory, the amygdala, which is the emotional memories, and adjacent cortex. When we say lobectomy, it is an operation which lobe is removed from the brain. So this picture, these are the medial temporal lobe that has been removed. Although his seizures were generally eliminated, partial seizures occur less 1 or 2 per day, and level of medication was reduced. HM bilateral medial temporal lobectomy was an unqualified success because it produces an amnestic effect where he had mild retrograde amnesia, which is unable to recall past events for those events that occurred in the 2 years before his surgery. HM also had an almost total inability to form a new long-term memories. Once he stopped thinking about new experience, it was lost forever. In effect of HM became suspended in time on the day in 1953 when he regained his health but lost his future. His family moved shortly after his surgery but he was never able to remember his new address or where commonly used items were kept in his new residence. He never learned to recognize people, like he doesn't know the doctors and the nurses, who he did not meet until after his surgery. And he read the same magazine over and over without finding them familiar. If you meet HM at a party, he could chat quite normally until he was distracted. For example, he was distracted by the phone, then he would not remember you. The conversation or where he was, it was as if he continually regained consciousness. Henry Molaison is the last patient to receive a bilateral medial temporal lobectomy because of its amnestic effect. In order to characterize HM anterograde memory problem, researchers began by measuring his performance on objective tests of various kinds of memory. This describes the 5 tests that were used to assess HM's long-term memory. The result of the first 2 tests, HM severe defects in long-term memory, whereas the result of the last 3 indicated that HM's brain was capable of storing long-term memories, but that HM had no conscious awareness of those memories. So 5 tests that were used to assess HM long-term memories are DigiSpan subset test, block tapping memory span test, mirror drawing test, incomplete pictures test, Pavlovian conditioning. So the first one is DigiSpan subset test. It's one of the main tools developed to measure one's verbal working memory, a classic test of verbal long-term memory. This test was used to objectively illustrate HM inability to form certain long-term memories. During the test of HM, he was asked to repeat 5 digits that were read to him at 1 second intervals. Like, for example, 1, 2, 3, 4, 5. This was repeated and he repeated the sequence correctly. On the next trial, the same 5 digit was presented in the same sequence. However, there was an additional digit added on the end. So it's become 6 digit already. So the same 6 digit sequence was presented a few times until he got it right. And then another digit was added to the end of it and so on. After 25 trials, HM had not managed to repeat 8-digit sequence, wherein most people can correctly repeat about 15 digits after 25 trials of this DigiSpan subset test. So the second one is we have the block tapping memory span test. It is a test to assess visual spatial working memory. So because of this test, researchers found out that HM had a global amnesia, which is a temporary episode of memory wherein your recall of recent events simply vanishes, so you can't remember where you are or how you got there. So how was black tapping memory span test was demonstrated? It was demonstrated by an array of 9 blocks that was spread out on a board in front of HM. And he was asked to watch the neuropsychologist to touch a sequence of them and then to repeat the same sequence of touches. So HM had a black tapping span of 5 blocks, which is in the normal range, but he could not learn to correctly touch a sequence of 6 blocks, even when the same sequence was repeated 12 times. So the third one is mirror drawing test. It is used to measure which thing rate of learning transfer of learning creativity. It is the first indication that HM anterograde amnesia did not involve all long-term memories came from result of mirror draw test. HM was asked to draw a line within the boundaries of a star-shaped target by watching his hand in a mirror. So you can see it in this picture. Like you just look at yourself drawing in the mirror. So HM was asked to trace the star 10 times on each of 3 consecutive days, and the number of times he went outside the boundaries on each try was recorded. As HM performance improved over the 3 days, which indicates retention of the task. However, despite of his improved performance, HM could not recall ever having completed the task before. Parang he thought that it was done for the first time. So the fourth one is the incomplete picture test. It is used as a measure of visual development as a clinical test for parietal cortex dysfunction and to examine long-term memory in amnestic patients. This was the discovery that HM was capable of forming long-term memories for mere drawing suggested that sensory-motor tasks were the one exceptions to his inability to form long-term memories. However, this view was challenged by the demonstration that HM could also form new long-term memories. So, how was this test done? The set contains drawings of the same 20 objects, but the set differ in their degree of completeness. Set 1 contains the most fragment drawings and set 5 contains the complete drawings. So like parang like what we use in the back when we were young, like connected dots is like the same thing, but the images are incomplete. The subject is asked to identify the 20 objects from the most fragmented set, which is set 1. Then those objects that go unrecognized are presented in their set 2 versions, and so on until all 20 items has been identified. As you can see, there is a performance of HM on this test. Here in this picture below, you can see that this illustrates the performance of HM on this test and his improved performance 1 hour later. Like he can connect all the images. However, despite of this improved performance, HM could not recall previously performing the task, like what he had experienced in the mirror drawing test. And lastly, we have the Pavlovian conditioning. Pavlovian conditioning is, we all are aware of this because it was discussed to us on our previous subjects. So it is a learning procedure in which a biologically potent stimulus is paired with a previously neutral stimulus. So HM learned an eye blink Pavlovian conditioning task at a retarded rate. A tone was sounded, which is the tone was the neutral stimulus, just before the puff of air, which is the unconditioned stimulus, was administered to his eye. These trials were repeated until the tone, which is now the conditioned stimulus, alone elicited an eye blink. So 2 years later, HM retained this conditioned response almost perfectly, although he had no conscious recollection of the training. So let's move on to the 3 major scientific contribution of HM case. Although HM case is a story of personal tragedy, this story has great contribution of the study of neural bases. So HM case shows that the medial temporal lobes play an especially important role in memory. In so doing, HM's case renewed efforts to relate individual brain structure to specific mnemonic or memory-related processes. In particular, HM case spawned a massive research effort aimed to clarify the mnemonic function of the hippocampus and other medial temporal lobe structure. So this is the first major scientific contribution of HMKs, that medial temporal lobes play an important role in memory. The second one is the discovery of the bilateral medial temporal lobectomy abolished HM's ability to form certain kinds of long-term memories without disrupting his performance on tests of short-term memory or his remote memory. Supported the theory that there are different modes of storage of short-term, long-term, and remote memory. HM's specific problem appeared to be difficult in memory consolidation, which is the translation of short-term memory into long-term memory. So HM has this kind of difficulty. It is first to reveal that an amnestic patient might claim no recollection of previous experience while demonstrating memory for it by improved performance. In short, memories may exist but not recalled. This discovery led to the creation of two distinct categories of long-term memories, which is the explicit memories or declarative memory, is the conscious intentional recollection of factual information, previous experiences, and concepts. We also have the implicit memories, referred to as an unconscious memory or automatic memory. It is used past experience to remember things without thinking about them. The performance of implicit memory is enabled by previous experiences, no matter how long ago those experiences occurred. Example of explicit and implicit memory. Explicit memory is recalling phone number, remembering what time you're meeting with a friend, while the example of implicit is that you can sing along to a familiar song and brushing teeth. So basically, that is the three major scientific contribution of HMKs. So let's move on to medial temporal lobe amnesia. Neuropsychological patients with a profile of mnemonic with preserved intellectual functioning and with evidence of medial temporal lobe damage are said to suffer from medial temporal lobe amnesia. It is a difficulty in forming explicit long-term memories while retaining the ability to form implicit long-term memories of the same experiences. This problem has proved to be a symptom of medial temporal lobe amnesia as well as many other amnestic disorders. Tests that assesses implicit memory are called repetition priming tests. Example of this is the incomplete picture test, when the participants are asked to examine the list of words. They're not asked to learn or remember anything. Later, they are shown series of fragments of words from the original list and are simply asked to complete them. Controls who have seen the original words perform well surprisingly. Participants with amnesia often perform equally well, even though they have no explicit memory of seeing the original list. Research on medial temporal lobe amnesia taught us assessment of implicit long-term memory now plays important role in the study of human brain. The problem with implicit and explicit memory has proved to be a symptom of medial temporal lobe amnesia as well as many other amnestic disorders. And discovered two memory systems, the implicit and explicit, which led to presumption that implicit system was the first to evolve because it is more simple. It does not involve unconsciousness. And after having two experiments, one with the amnestic patient and one with the amnestic monkey, with a major temporal lobe lesion, suggests that the answer is flexibility. In both experiments, the amnestic subjects learned the implicit learning task as well as the control subject did presumably The evolution of explicit memory system provided for the flexible use of information. So next is semantic and episodic memories. Study found that explicit memories fall into two categories. We have the semantic memory and the episodic memory. Semantic memory are explicit memories for general facts or information, includes things that are common knowledge such as the name of colors, the sound of letters, the capitals of countries, and the other basic facts acquired over a lifetime. On the other hand, episodic memory is an explicit memory for the particular events, situation, and experiences of one's life. Examples are memory of your first day of school, your first love, attending a friend's birthday party. People with medial temporal lobe amnesia have particular difficulty with episodic memories. And Endeltovic has been a major force in research on the semantic episodic dichotomy. Following is the case of his patient, Casey. Casey's episodic memory, also known as autobiographical memory, where he has been likened to traveling back in the time mentally and experiencing one's past. So Casey's case was he had a motor accident and suffered diffuse brain damage, including damage to the medial temporal lobes. His cognitive ability remained normal, where he can play organ, chess, and other various games. Still, in the midst of these normal memories, Casey has severe amnesia for personal experiences, so he cannot recall a single personal event for more than a minute or two. This is an inability to recall any episode, events at which he was present covers his entire life. In his case, he has no difficulty having a conversation, unlike some people who has amnesia, since he does use his semantic memory. However, Casey understands time but cannot time travel into either the past or the future. He cannot imagine his future any better than he can recall his past. He can't imagine what he will be doing for the rest of the day, the week, or his life. In the book, there is also another case about episodic memory defect wherein a clever neuropsychologist spotting episodic memory defect, since it is difficult to spot episodic defect because neuropsychologist has no idea of the true events of the patient's life. And some part is that the patient provides effective semantic answers to episodic questions. So if you are interested on how did this clever neuropsychologist spotted the episodic memory defect of his or her patient, you can read it on the book. It is interesting as well. The next is effects of global cerebral ischemia on the hippocampus and memory. Patients who have experienced global cerebral ischemia have experienced the interruption of blood supply to their entire brains, often suffer from medial temporal lobe amnesia. RB is one such individual. So the case of RB is a product of a bungled operation. At the age of 52, RB underwent cardiac bypass surgery. The surgery was bungled and as a consequence, RB suffered brain damage. So usually, if you went under cardiac bypass surgery through the ischemia, there are high chances that you can suffer from brain damage. So the pump that was circulating an RB's blood to his body while his heart was disconnected broke down, and it was several minutes before a replacement arrived from another part of the hospital. RB lived, but the resulting ischemic brain damage left him amnesic. RBK suggests that hippocampal damage by itself can produce medial temporal lobe amnesia. However, in such cases of cerebral ischemia, it is difficult to rule out the possibility of subtle damage to other areas of the brain. The strongest evidence that selective hippocampal damage can cause medial temporal lobe amnesia comes from the cases of transient global amnesia. So what is transient global amnesia? Transient global amnesia is defined by its sudden onset in the absence of any obvious cause in otherwise normal adults. It is a sudden temporary episode of memory loss that can't be attributed to a more common neurological condition such as epilepsy or stroke. During an episode of transient global amnesia, your recall of recent events simply vanishes, so you can't remember where you are, how you get there. So transient global amnesia is really scary thing, especially if this is an onset transient global amnesia, since you can never know when will this occur, and if that will occur, you will be scared of that moment. So overall effect of global cerebral ischemia on the hippocampus and memory is the abnormality of CA or homoammonis, which is hippocampus subfields, wherein total number of CA neurons are reduced. Hippocampal damage can produce medial temporal lobe amnesia and comes from cases of transient global amnesia. So let's move on to amnesia of Karaskov syndrome. Karaskov syndrome was already discussed by chapter 5, I think. It was already discussed, but anyways, I'm gonna discuss it again. Karaskov syndrome is a disorder of memory common in people who have consumed large amounts of alcohol. The disorder is largely attributable to the brain damage associated with the thiamine deficiency that often accompanies heavy alcohol consumption. So Karaskov syndrome usually is caused by thiamine deficiency or heavy alcohol consumption. In its advanced stages, it is characterized by a variety of sensory and motor problems, extreme confusion, personality changes, and a risk of death from liver, gastrointestinal, or heart disorders. Postmortem examination typically reveals lesion to the medial diencephalon, the medial thalamus, and the medial hypothalamus, and diffuse damage to several other brain structures, most notably to neocortex, hippocampus, and cerebellum. The amnesia of Korsakoff syndrome is similar to medial temporal lobe amnesia in some respects. The gradual, insidious onset and progressive development of Korsakoff syndrome complicate the study of the resulting retrograde amnesia. Because the brain damage associated with Korsakoff syndrome is diffuse, it has not been easy to identify which part of it is specially responsible of amnesia. The first hypothesis, which was based on several small post-mortem studies, was that damage of the mammillary bodies of the hypothalamus was responsible for the memory defects of Korsakoff patients. However, subsequent studies revealed cases of Korsakoff amnesia with no mammillary body damage. But there was damage to another pair of medial diencephalic nuclei, the medial dorsal nuclei of thalamus. NA is particularly well-known patient with medial diencephalic amnesia, amnesia such as Korsakoff amnesia associated with the damage of the medial diencephalon. Although his memory defects were conventional, their causes was not. The last for my report is amnesia of Alzheimer's disease. Alzheimer's disease is another major cause of amnesia. It is an irreversible progressive brain disorder that slowly destroys memory and thinking skills and eventually the ability to carry out the simplest task. In most people with this disease, those with the last onset type symptoms first appear in their mid-60s. So usually Alzheimer's disease occurs on mid-60s people. The first sign of Alzheimer's disease is often a mild deterioration of memory. However, the disorder is progressive. Eventually, dementia develops and becomes so severe that the patient is incapable of even simple activities such as eating, speaking, recognizing a spouse, or bladder control. So if you can watch, there are a lot of movies with people who have Alzheimer's disease, and usually this these activities like they can't eat, they can't speak well, they can't recognize their spouse, and the bladder control, like they will just they will need a diaper for that. So Alzheimer's disease is terminal. The neural basis of Alzheimer amnesia has focused on pre-dementia Alzheimer patient. Pre-dementia Alzheimer patient are Alzheimer patients who have yet to develop dementia. The memory defects of these patients are more general than those associated with medial temporal lobe damage, medial diencephalic damage, or Korsakoff syndrome. Patients often display deficits in short-term memories and in some types of implicit memory. So, what is the etiology of Alzheimer? Alzheimer's disease is thought to be caused by the abnormal buildup of proteins in around brain cells. One of the proteins involved is called amyloid deposits, of which form plaques around brain cells. The other protein is called tau deposits, of which form tangles within the brain cell. So, although it's not known exactly what causes this process to begin, scientists now that know that it begins many years before symptoms appear. So Alzheimer's disease are already forming a long time ago. So this picture here is how our normal brains look like. And this is the brain of patients with Alzheimer's disease. Parang it started to shrink. So the first symptoms of Alzheimer's disease is a problem with vision, language, rather than memory. So, fun fact, nauna muna ang vision or language rather than memory. So, here are some signs of amnesia. So, for mild Alzheimer's disease, we have memory loss, poor judgment leading to bad decisions, loss of spontaneity and sense of initiative, taking longer to complete normal daily tasks, repeating questions, trouble handling money or paying bills, wandering and getting lost, losing things or misplacing them in odd places, mood and personality changes, increased anxiety and or aggression. For moderate Alzheimer's disease, these are the signs: increased memory loss and confusion, inability to learn new things, difficulty with language and problems with reading, writing, and working with numbers. Difficulty organizing thoughts and thinking logically, shortened attention span. Problems coping with new situation. Difficulty carrying out multi-step tasks such as getting dressed. Problem recognizing family and friends. Hallucination, delusion, and paranoia. Impulse behavior such as undressing at inappropriate times or place or using vulgar language, inappropriate outbursts of anger, restlessness, agitation, anxiety, tearfulness, wandering, especially in the late afternoon or evening, repetitive statements or movement, occasionally muscle twitches. And last is the severe Alzheimer's disease, which is inability to communicate, like your brain is already deteriorating, like you can communicate, you lose weight, you can produce seizures, skin infection, difficulty swallowing, groaning, moaning, or grunting, increased sleeping, loss of bowel and bladder control. Basically, that is all for my report. So I do hope that you did learn just a little bit learning. So before we move on to our next reporter, let us first test ourselves. So number 1, please comment down your answers. I'll be watching you guys. So number 1, blank are explicit memories for general facts or information. So I'll be giving you 30 seconds to answer this. Comment now. Okay, time is running out. So the correct answer is semantic memory. Number 2, blank explicit memories for the particular events of one's life. Timer starts now. The correct answer is episodic memory. Number 3, blank refers to a learning procedure in which a biologically potent stimulus is paired with a previously neutral stimulus. Timer starts now. So the correct answer is Pavlovian conditioning. Number 4, blank is the conscious intentional recollection of factual information, previous experiences, and concepts. The answer is explicit memories or declarative memory. Lastly, blank referred to as unconscious memory or automatic memory. It uses past experiences to remember things without thinking about them. The answer is implicit memories.
Good morning, everyone. Hello. I'm here to talk about lessons 11.8 to 11.14. Before I get started, I would like to apologize for the glare in my glasses whenever I move my head a certain way, and some background noises that may come up. I would like to apologize for that as it can be distracting. Anyway, let's get started. Uh, Lesson 11.8, Post-Traumatic Amnesia. Sometimes people get hit in the head. It's not hard enough to kill them, but it is hard enough to discombobulate them. Concussion is a temporary disturbance of consciousness caused by a non-penetrating head injury. Post-traumatic amnesia, or PTA, is an amnesia following a non-penetrating blow to the head. A coma is a pathological state of unconsciousness after a severe blow to the head. It can last between a few seconds to a few years. When a patient wakes up from a coma, they experience a period of confusion. Tests will have to be taken to see if the patients are suffering with amnesia. There are two types of amnesia. First is retrograde amnesia, or the type of amnesia wherein you are unable to remember what happened in the years or even decades prior to the injury. This type of amnesia is more known as it is more commonly portrayed in media, like in Filipino teleseries, it's almost a cliche. And anterograde amnesia refers to the decreased ability to retain new information. This type of amnesia is also portrayed in media, but less. This is the type of amnesia featured in the movie called 50 First Dates, if I remember correctly, where the girl spends the same day over and over again because she can't retain new memories. She can't make new memories, so she lives the same day over and over again. More severe blows to the head tend to produce longer comas, longer periods of confusion, and longer periods of amnesia. Now, if you look to the picture down here, you can see that there are stages. First stage is a blow to the head that produces the coma. Second stage is when a victim regains consciousness and there is a period of confusion. And the third stage is when a period of confusion ends and the victim has retrograde amnesia for the events that occurred during the period just before the blow, and anterograde amnesia for events that occurred during the period of confusion. Lesson 11.9, Gradients of Retrograde Amnesia and Memory Consolidation. Gradients of retrograde amnesia seem to provide evidence for memory consolidation, which we will further discuss now. Hebb, who we know from chapter 1, is back with a theory, a brain theory. Okay, that wasn't funny. Anyway, Heb argues that memories of experiences are stored in the short term by neural activities reverberating in closed circuits. These patterns are susceptible to disruption, but eventually they introduce structural changes in the synapse, which provide stable long term storage. Now, how do scientists replicate uh, neural activity disruption? Scientists use ECS, or electroconvulsive shock. This is an intense, brief seizure inducing current that is administered to the scalp. The rationale for using ECS to study memory consolidation was that by disrupting neural activity, ECS would erase from the storage only those memories that had not yet been converted to structural synaptic changes. The length of the period of retrograde amnesia produced by an ECS would thus provide an estimate of the amount of time needed for memory consolidation. The current view of memory consolidation is that it continues for a long time, if not indefinitely. Hippocampus and consolidation. The case of HM, which was discussed earlier, provided evidence of memory consolidation, and it seemed to suggest that the hippocampus played a special role in it. Standard consolidation theory, or the dual trace theory. Scoville and Milner in 1975 suggested that memories are temporarily stored in the hippocampus until they can be transferred to a more stable cortical storage system. This is to account for the fact that the bilateral medial lobectomy disrupted only those retrograde memories in the few years before HM's surgery. Adherence to the standard consolidation theory. It seems unlikely that the hippocampus plays a special role in consolidation. It happens that when a conscious experience occurs, it is rapidly and sparsely encoded in a distributed fashion through the hippocampus and other involved structures. According to Nadel and Moscovich, retained memories become progressively more resistant to disruption by hippocampal damage because each time a similar experience occurs and the original memory is recalled, a new engram is established and linked to the original engram, making the memory more easier to recall and the original engram more difficult to disrupt. Now, what is this engram that we mentioned? An engram is the change in the brain that stores a memory. Reconsolidation. A hypothesis that each time a memory is retrieved from a long-term storage, it is temporarily held in a labile short-term memory where it is once again susceptible to post-traumatic amnesia until reconsolidated. Labile meaning changeable or unstable. Okay. Lesson 11.10. Animal models of object recognition amnesia, the delayed non-matching to sample test. In this test, in the delayed non-matching to sample test, a monkey is presented with a distinctive object, or the sample object, under which it finds food, and then after a delay, the monkey is presented with two test objects, the sample object and an unfamiliar object. The monkey must remember the sample object so it can select the unfamiliar object to obtain food concealed beneath it. So here are the results for that. Intact, well-trained monkeys perform correctly about 90% of the delayed non-matching sample trials, when the retention intervals were a few minutes or less. So the monkeys without lesions or the intact monkeys did well and passed with flying colors about 90% of the time. However, the monkeys with bilateral medial temporal lobe lesions had major object recognition, meaning their results weren't as good, but their results mirrored that of HM in some respects. Humans with medial temporal lobe amnesia have been tested on the delayed non-matching sample test. Their rewards were coins rather than banana slices, and their performance mirrored that of monkeys with the similar brain damage. The development of delayed non-matching sample tests for monkeys provide a means of testing the assumption that amnesia resulting from the medial temporal lobe damage is entirely a consequence of the hippocampal damage. Now, if you look at the chart, you will see that the people with the subjects with medial temporal lobe lesions, represented in blue, actually do well in 8-second delays, but the longer you delay it, the lower their scores get, meaning they're kind of, they get worse and worse the longer the delays go. But the people with normal controls, the one with uh, no lobe lesions, have consistent, if not better, results. Okay, so the rat version of the delayed non-matching sample test. Because of the size and location of the hippocampus, almost all studies of hippocampal lesions in monkeys have involved aspiration or suction of large portions of the medial temporal cortex in addition to the hippocampus. So they tried it in rats because they have smaller brains. In rats, the extraneous damage associated with aspiration lesions of the hippocampus is typically limited to a small area of parietal neocortex. Furthermore, the rat hippocampus is small enough that it can be lesioned electrolytically or with intracerebral neurotoxin injections, which are methods that produce less extraneous damage, therefore it has less suction, or if not, there is no suction. Okay, so now we will be going to the Mumby box. The Mumby box is a version of the delayed non-matching sample test for rats that most closely resembles that for monkeys, which is developed by David Mumby. That's why it's called the Mumby box. The validity of the rat version of the delayed non-matching sample test has been established by studies of the effects of medial temporal lobe lesions. As in humans and monkeys, bilateral lesions of rats, hippocampus, amygdala, and medial temporal cortex combined, Jesus, produce major deficits at all but the shortest retention intervals. So their results are just like monkeys and people's. The longer the intervals, the worse they get. In the early 1990s, researchers began assessing relative effects of lesions to the various medial temporal lobe structures on performance in the delayed non-matching sample test in both monkeys and rats. Challenges to the view that hippocampal damage is the critical factor in medial temporal amnesia quickly accumulated. Most reviews of this research reached similar conclusions. Bilateral surgical removal of the medial temporal cortex consistently produces severe and permanent deficits in performance on the delayed non-matching the sample test and other tests of object recognition. In contrast, bilateral surgical removal of the hippocampus produce only modest deficits, and bilateral destruction of the amygdala produces none. So if we look at this graph, in the medial temporal cortex lesions here, over here below, we can see that the medial temporal cortex lesions, they did well. The subjects did well pre-surgery, but after the surgery, just like before, the longer the intervals, the worse their scores get. However, in the hippocampus plus amygdala lesions, their deficits are more modest, less worse than before, but it is still evident. But it's not as evident. Okay, so these are some devices used in rats to test the deficits in bilateral medial temporal lobectomy. First is the Morris water maze test. Intact rats placed at various locations in a circular pool of murky water rapidly learn to swim in a stationary platform hidden just below the surface. Rats with hippocampal lesions learn this simple task with great difficulty. Next is the radial arm maze test. The radial arm maze test is a test where several, in this case 8 arms radiate out of a central starting chamber and the same few arms are baited with food each day. Intact rats readily learn to visit only those arms that contain food and do not visit the same arm more than once a day. Rats with hippocampal lesions display major deficits on both the reference memory and the working memory measures of radial arm maze performance. The ability for rats to be able to visit the baited arms is a measure of reference memory. Reference memory is memory for the general principles and skills that are required to perform a task, and the ability to refrain from visiting an arm more than once in a given day is a measure of working memory, or the temporary memory that is necessary for the successful performance of a task on which one is currently working. Lesson 11.12, Hippocampal place cells and entorhinal grid cells. Place cells are neurons that respond only when a subject is in specific locations. Entorhinal cortex is an area of the medial temporal cortex that is a major source of neural signals to the hippocampus. Grid cells are entorhinal neurons that each have an extensive array of evenly spaced place fields, producing a pattern reminiscent to graph paper. Other types of neurons in the entorhinal cortex are associated with spatial location. For example, head direction cells turn to the direction of the head orientation, and border cells fire when the subject is near the borders of its immediate environment. The nature of the relationship between entorhinal grid cells and hippocampal place cells is still a matter of ongoing debate. First, there is a major pathway from the entorhinal cortex to the hippocampus. Second, entorhinal grid cells respond relatively reflexively to location, whereas hippocampal place cells respond to the place in combination with other features of the test environment. The discovery that the properties of hippocampal place cells emerge in developing rat pups prior to the emergence of stable entorhinal grid cell firing, challenges the idea that input from entorhinal grid cells is essentially for hippocampal place cell function. Moreover, there is evidence that place cells can still function after entorhinal grid cells have been eliminated. Lesson 11.13, Comparative Study of the Hippocampus and Spatial Memory. Although research on a variety of species indicate that the hippocampus does play a role in spatial memory, the effects of hippocampal damage on the performance of spatial memory tasks in humans and other primates have been less consistent. One problem may be that spatial memory in humans and monkeys is often tested while they remain stationary and make judgments of locations on computer screen, whereas spatial memory in rats, mice, and birds is typically studied as they navigate through test environments. Lesson 11.14, Jennifer Aniston cells, neurons, concept cells. Okay. Jennifer Aniston neurons are highly selective. Each neuron responded only to a small number of test objects or individuals. Often only one could be found. Also, their responses are highly invariant. If a neuron responded to a particular object on test 1, it tended to respond to that object in all subsequent tests. Now, a few of you may be wondering, why is it called Jennifer Aniston? Isn't Jennifer Aniston in Friends? Well, the answer to that question, my dear classmates, is because the first neurons to be studied this way fired in response to an image of Miss Jennifer Aniston. That's why it's called that. Without question, the most remarkable feature of Jennifer Aniston neurons is that they respond to ideas or concepts rather than to particulars, which is why they are also known as concept cells. Interestingly, when Jennifer Aniston neurons or concept cells have been found to respond to more than one concept, there is usually an obvious relation between them. And with that, we reach the end of lesson 11.14. Next thing that we're going to do is test your brains. I'm going to show you the question and I'm going to give you 30 seconds to answer it in the comments below. Are y'all ready? Okay. Okay. So first question. Okay, there we go. First question. A blank is a pathological state of unconsciousness after a severe blow to the head. It can last between a few seconds to a few years. The timer starts now. Okay, the answer is coma. A coma is a pathological state of unconsciousness after a severe blow to the head. It can last in between a few seconds to a few years. Please ignore that, although in some instances I forgot to erase that. Okay, next. Blank refers to a decreased ability to retain new information. Timer starts now. Time's up. The answer is anterograde amnesia. Anterograde amnesia refers to a decreased ability to learn new information. Next question. Blank is a change in the brain that stores a memory. Timer starts now. Time's up. The answer is engram. Engram is a change in the brain that stores a memory. Next question. Blank are neurons that respond only when a subject is in specific locations. Timer starts now. Time's up. The answer is place cells. Place cells are neurons that respond only when a subject is in specific locations. Last question. The version of the delayed non-matching to sample tests for rats that most closely resembles that for monkeys, developed by David Mumby. Timer starts now. Time's up. The answer is Mumby box, the version of the delayed non-matching to sample test for rats that most closely resembles that for monkeys, developed by David Mumby, is called the Mumby box. I am back. That is the end of my report. I am going to pass the floor, the mic, to Miss Angela Amoylin. Take it away.
As you learned in Chapter 5, one new approach in the toolkit available to biopsychologists is the optogenetics. So what is optogenetics again? As you recall, optogenetics most commonly refers to a biological technique that involves the use of light to control neurons that have been genetically modified to express light-sensitive ion channels. So in recent years, this tool has been used extensively in studies of learning and memory in mice, according to Goshen 2014. One line of research has been particularly interesting because it can shed light on the location of neurons that maintain an engram. So engram cells. What What are engram cells? So engram cells are a population of neurons that are activated by learning, have enduring cellular changes as a consequence of learning, and whose reactivation by a part of the original stimuli delivered during learning results in memory recall. Engram cells were discovered through optogenetics. The identification of engram cell via optogenetics is typically a two-stage process. So the first process is the tagging stage. This is where the neurons that are active during the learning task are induced to express opsins while an animal engages in a particular learning task. And the next process or the next stage is the manipulate stage. The, the previously active neurons are now either inhibited or excited by using light to influence the activity of the opsin tag neuron. Through optogenetics, researchers are now able to observe, suppress, or activate engram cells in different parts of the nervous system. For example, researchers have been able to reverse depressive-like behavior in mice by optogenetically reactivating hippocampal dentate granule cells that had previously been active during the encoding of a positive experience. Moreover, researchers have shown that in transgenic mouse models of Alzheimer's disease, activating engram cells leads to retrieval of memories that are otherwise inaccessible, suggesting that the memory deficits of Alzheimer's disease are retrieval deficits rather than encoding deficits. Have you ever wondered where our memories are stored? According to Lashley, if a particular structure were the storage site for all memories of particular type, then destruction of that structure should eliminate all memories of that type that were acquired prior to lesions. No brain structure has shown this result. Lesions of particular structures tend to produce either no retrograde amnesia at all or retrograde amnesia for only the experiences that occurred in the days or weeks just before the surgery. So these findings of Lashley have led to two major conclusions. First is that memories are stored diffusely in the brain and thus can survive destruction of any single structure. The second is that memories become more resistant to disruption over time. Aside from these neural structures the, that appear to play some role in the storage of memories, the hippocampus, the medial temporal cortex, the mediodorsal nucleus of the thalamus, and the basal forebrain, there are also another five brain areas implicated in memory: the inferotemporal cortex, amygdala, prefrontal cortex, cerebellum, and the striatum. So the first is the inferotemporal cortex. Um, Areas of the brain that are active during the retention of an experience tend to be the same ones active during the original experience. This has focused attention on the mnemonic functions of the sensory and motor areas of the brain. In particular, attention is focused on inferotemporal cortex. So, inferotemporal cortex or cortex of the inferotemporal cortex has complex visual functions. Its important role is to store memories of visual input. In support of this view, Naya, Yoshida, and Miyashita recorded the responses of neurons in inferotemporal cortex and perirenal cortex, while monkeys learned the relation between the two items in pairs of visual images. When a pair was presented, responses were first recorded in inferotemporal neurons and then in perirenal neurons. However, when the monkeys were required to recall that pair, activity was recorded in perirenal neurons before inferotemporal neurons. Nye and colleagues concluded that this reversed pattern of activity reflected the retrieval of visual memories from inferotemporal cortex. Next is the amygdala. The, the amygdala is thought to play a special role in memory for the emotional significance of experiences. In support of this view, rats with amygdalar lesions, unlike intact rats, do not respond with fear to a neutral stimulus that has previously been followed by electric foot shock. Also, um, Becerra and colleagues reported the case of neuropsychological patient with bilateral damage to the amygdala who could not acquire conditioned autonomic startle responses to various visual or auditory stimuli but had good explicit memory for the training. However, there is little evidence that the amygdala stores memories. It appears to be involved in strengthening emotionally significant memories stored in other structures. The amygdala the amygdala might be the reason why emotion-provoking events are remembered better than neutral events. So this gave me a thought that sometimes it's not the memory of what you saw or heard before that hurts, but the memory of pain you felt that time. Maybe. I'm not sure. Next is the prefrontal cortex. Prefrontal cortex, the area of frontal cortex anterior to the motor cortex. Um, Patients with damage to the prefrontal cortex are not grossly amnesic. They often display no deficit at all on conventional tests of memory. This lack of reliable memory deficits in patients with prefrontal damage may in part result from the fact that different parts of the prefrontal cortex play different roles in memory, and that patients with damage to different areas of prefrontal cortex are often combined for analysis. Lesions in prefrontal cortex also displays deficits in working memory. So what is working memory? Working memory is the ability to maintain relevant memories while a task is being completed. As a result of this deficit, patients with prefrontal cortex damage often have difficulty performing tasks that involve a series of responses. The prefrontal cortex is a large structure that is composed of many anatomically distinct areas that have different connections and presumably different functions. Functional brain imaging studies are finding that specific complex patterns of prefrontal activity are associated with various memory functions. Some regions of the prefrontal cortex seem to perform fundamental cognitive processes during work memory tasks, and other regions of prefrontal cortex participate in other memory processes. The case of the cookbook. The story of one patient with prefrontal cortex damage is very well known because she was the sister of Wilder Penfield, the famous Montreal neurosurgeon. Before her brain damage, she had been an excellent cook, and afterward she still remembered her favorite recipes and how to perform each individual cooking technique. However, she was incapable of preparing even simple meals because she could not carry out the various steps in proper sequence. Uh, so another areas implicated in memory are the cerebellum and the striatum. Just as explicit memories of experiences are presumed to be stored in the circuits of the brain that mediated their original perception, implicit memories of sensory-motor learning are presumed to be stored in a sensory-motor circuit. Most research on the neural mechanisms in memory or sensory-motor tasks has focused on two structures, the cerebellum and the striatum. So the cerebellum is thought to participate in the storage of memories of learned sensory-motor skills. In support of this view, Pavlovian conducted the conditioning of the eye blink response of rabbits. So the tone, which is the conditional stimulus, is sounded just before a puff of an ear, which is the unconditional stimulus, is delivered to the eye. After several trials, the tone comes to elicit an eye blink. The convergence of evidence from stimulation recording and lesion studies suggests that the effects of this conditioning are stored in the form of changes in the way of the cerebellar neurons respond to the tone. The striatum is thought to store memories for consistent relationship between stimuli and responses, the type of memories that develop incrementally over many trials. Sometimes this form of learning is referred to as habit formation. Although few would disagree that the cerebellum and the striatum play a role in sensory-motor memory, there is a growing evidence that these structures also play a role in certain types of memory with no obvious motor component. For example, Knowlton and Coolidge found that the Parkinson's patients who had striatal damage could not solve a probabilistic discrimination problem. The problem was a computer weather forecasting game, and the task was to correctly predict the weather by pressing one of two keys, rain or shine. The patients based their predictions on stimulus cards presented on the screen. Each card had a different probability of leading to sunshine, which the patients had to learn and remember. The Parkinson's patients did not improve over 50 trials, although they displayed normal explicit, conscious memory for the training episodes. In contrast, amnesic patients with medial temporal lobe or medial diencephalic damage displayed marked improvement in performance but had no explicit memory of their training. So we are done in the particular structures of the human brain that are involved in learning and memory and on what happens when these structures are damaged. Now we go to the synaptic mechanism of learning and memory. So most modern thinking about the neural mechanism of memory began with Hebb. Hebb argued so convincingly that enduring changes in the efficiency of synaptic transmission were the basis of long-term memory that the search for the neural basis of learning and memory has focused almost exclusively on the synapse. Because Hebb's hypothesis that enduring facilitations of synaptic transmission are the neural basis of learning and memory was so influential, there was a great excitement when such an effect was discovered. In 1973, Bliss and Lomo showed that there is a facilitation of synaptic transmission following high-frequency electrical stimulation applied to presynaptic neurons. This phenomenon this phenomenon is called the long-term potentiation, or the LTP. So LTP has been demonstrated in many species and in many parts of their brains, but it has been most frequently studied in the rodent hippocampus. Okay, so figure 11.9. Figure 1119 illustrates LTP in granule cell layer of the rat hippocampal dentate gyrus. First, a single low-intensity pulse of a current was delivered to perforant path, the major input to the dentate gyrus, and the response was recorded through the extracellular multiple unit electrode in the granule cell layer of the hippocampal dentate gyrus. The purpose of this initial stimulation was to determine the initial response baseline. Second, high-intensity, high-frequency stimulation lasting 10 seconds was delivered to the perforant path to induce the LTP. Third, the granule cell's responses to single pulses of low-intensity current were measured again after various delays. So, in figure 1119, it shows that transmission at granule cell synapses was still potentiated 1 week after the high-frequency stimulation. LP is among the most widely studied neuroscientific phenomena. Why? The reason goes back to 1949 and Hebb's influential theory of memory. The synaptic changes that have hypothesized as underlying long-term memory seem to be the same kind of changes that underlie LTP. So LTP has two key properties that have proposed as characteristics of the physiological mechanism of learning and memory. First is that LTP can last for a long time, for several months after multiple high-frequency stimulation, according to Abraham 2006. Second is that LTP develops only if the firing of the presynaptic neuron is followed by the firing of the postsynaptic neuron. It does not develop when the presynaptic neuron fires and the postsynaptic neuron does not. It does not develop when the if it does not develop when the presynaptic neuron does not fire and the postsynaptic neuron does. The co-occurrence of firing in presynaptic and postsynaptic cells is now recognized as the critical factor in LTP, and the assumption that the co-occurrence is a physiological necessity for learning and memory is often referred to as Hebb's postulate for learning. Additional support for the idea that LTP is related to the neural mechanisms of learning and memory has come from several observations. LTP can be elicited by the low levels of stimulation that mimic normal neural activity. Second is that LTP effects are most prominent in structures that have been implicated in learning and memory, such as the hippocampus. Learning can produce LTP-like changes in the hippocampus. Many drugs that influence learning and memory have parallel effects on LTP. The induction of maximal LTP blocks the learning of a Morris water maze until the LTP has subsided. Behavioral changes that appear to be memories can be induced in mice via LTP. And lastly, LTP occurs at specific synapses that have been shown to participate in learning and memory in simple invertebrate nervous system. But still, it is important to keep in mind that all of this evidence is indirect and that LTP as induced in the laboratory by electrical stimulation is the best a caricature of the subtle cellular events that underlie learning and memory. Conceiving of LTP as three-part process, many researchers are investigating the mechanisms of induction, maintenance, and expression. That is, the process by which high-frequency stimulation induce LTP, or learning. The changes responsible for the maintenance of LTP, or memory, and the changes that allow it to be expressed during the test or recall. The NMDA or N-methyl D-aspartate receptor is prominent at the synapses at which LTP is commonly studied. The NMDA receptor is a receptor for glutamate. As we recall in Chapter 4, glutamate is the main excitatory neurotransmitter of the brain. So an NMDA receptor does not respond maximally unless two events occur simultaneously. Glutamate must bind to it, and the postsynaptic neuron must already be partially depolarized. This dual requirement stems from the fact that the calcium channels associated with NMDA receptor allow only small numbers of calcium ions to enter the neuron unless the neuron is already depolarized. When glutamate binds to the receptor, it is the influx of calcium ions that triggers the cascade of events in the postsynaptic neuron that induces LTP. An important characteristic of the induction of LTP at glutaminergic synapses stems from the nature of NMDA receptor and their requirement for co-occurrence of firing for LTP to occur. This characteristic is not obvious under the usual but unnatural experimental condition in which LTP is induced by high-intensity, high-frequency stimulation which always activates the postsynaptic neurons through massive temporal and spatial summation. However, when a more natural low-intensity stimulation is applied, the postsynaptic neurons do not fire, and thus LTP is not induced, unless the postsynaptic neurons are already partially depolarized so that their calcium channels open wide when glutamate binds to their NMDA receptors. The requirement for the postsynaptic neurons to be partially depolarized when the glutamate binds to the NMDA receptor is an extremely important characteristic of conventional LTP because it permits neural networks to learn associations. As you can see in figure 11:20, the induction of NMDA receptor mediated LTP. So if one glutaminergic neuron were to fire by itself and release its glutamate neurotransmitter across a synapse onto the NMDA receptors of a postsynaptic neuron, there would be no potentiation of transmission at that synapse because the postsynaptic cell would not fire. However, if the postsynaptic neuron were partially depolarized by input from other neurons when the presynaptic neuron fired, the binding of the glutamate to the NMDA receptors would open wide the calcium channels. Calcium ions would flow into the postsynaptic neuron and transmission across the synapse between the presynaptic and postsynaptic neuron would be potentiated. The requirement for co-occurrence and the dependence of NMDA receptor on simultaneous binding and partial depolarization mean that under natural conditions, synaptic facilitation records the fact that there has been simultaneous activity in at least two converging inputs to the postsynaptic neuron, as would be produced, for example, by the simultaneous presentation of conditional stimulus and an unconditional stimulus. Maintenance and expression of LTP storage and recall. The search for the mechanism underlying the maintenance and expression of LTP began with attempts to determine whether these mechanisms occur in presynaptic or postsynaptic neurons. However, this question has been answered already. The maintenance and expression of LTP involve changes in both presynaptic and postsynaptic neurons. This discovery indicated that the mechanism underlying the maintenance expression of LTP are complex. After more than 4 decades of research, we still do not have satisfactory answers. However, the quest for the neural mechanism of LTP maintenance and expression has contributed to several important discoveries. So, here are the three of them. First is that once it became apparent that LTP occurs only at specific synapses on a postsynaptic neuron, it was clearly that there must be a mechanism for keeping the events at one set of synapses on a postsynaptic neuron from affecting other synapses on the same neuron. This specificity is due to the dendritic spine. So dendritic spines refers to the calcium ions that enter a dendritic spine that do not readily pass out of it. Thus, they exert their effects locally. Next, it became apparent that maintenance of LTP involves structural changes which depend on protein synthesis. The discovery that LTP causes structural changes was exciting because the structure of neurons and neural circuits had been assumed to be static. Many kinds of structural changes have been described, and the changes have turned out to occur for more rapidly and more frequently than was once assumed. And lastly, the discovery of structural changes in neurons following the induction of LTP stimulated a search for a mechanism by which neurons' activity could change its structure. This led to the discovery of numerous transcription factors that were activated by neural activity. So, transcription factors again refers to the intracellular proteins that bind to DNA and influence the operation of particular genes. Most of the research on LTP has focused on NMDA receptor-mediated LTP in the hippocampus. It is now clear that NMDA receptor-mediated LTP involves a complex array of changes that are difficult to sort out. In addition, LTP has been documented in many other parts of the CNS, where it tends to be mediated by different mechanisms. So there is LTD or long-term depression. LTD is the flip side of LTP and occurs in response to prolonged low-frequency stimulation of presynaptic neurons. And metaplasticity refers to the fact that the LTP and/or LTD induction can be modulated by prior synaptic activity. We now move on to infantile amnesia. So, we all experience infantile amnesia. That is, we remember virtually nothing of the events of our infancy. Newcomb and her colleagues address some questions like Do normal children who fail to explicitly recall or recognize things from their early childhood display preserved implicit memories for these things? The result of two experiments indicate that the answer is yes. In one study of infantile amnesia, children were shown a series of photographs of preschool-aged children, some of whom had been their preschool classmates. The children recognized a few of their former preschool classmates. However, whether they explicitly remembered a former classmate or not, they consistently displayed a large skin conductance response to the photographs of their former classmates, but not to the control photographs. In a second study of infantile amnesia, Drummie and Newcomb used a version of incomplete pictures test. First, they showed a series of drawings to 3-year-olds, 5-year-olds, and adults. 3 months later, the researchers assessed the implicit memories for these drawings by asking each participant to identify the drawings. For example, they showed a car, a drawing of a car and a chair, and some controlled drawings as quickly as they could. During the test, the drawings were first presented badly out of focus, but became progressively sharper over time. Following this test of implicit memory, explicit memory was assessed by asking the participants which of the drawings they remembered seeing before. The 5-year-olds and adults showed better explicit memory than 3-year-olds did. That is, they were more likely to recall seeing drawings from the original series. However, all three groups displayed substantial implicit memory. In conclusion, all participants were able to identify the drawings they had previously seen sooner, even when they had no conscious recollection of them. Smart drugs. Interesting. As students, most of us struggle in memorizing things, diba? So, who among you here uses smart drugs? Do they work? Do they really work? They're real, but do they really work? So, smart drugs are nootropics. Nootropics are substances that are thought to improve memory. These drugs are commonly seen on the internet, food stores, TV, advertisement like that. Those offering nootropics for sale claim that scientific evidence have proven that these substances improve the memories of healthy children and adults and block the adverse effects of aging on memory. But are these claims really supported by valid scientific evidences? The evidence that nootropics enhance memory has been reviewed several times by independent scientists. So the following pattern of conclusions has emerged from these reviews. First, although nootropics are often marketed to healthy adults, most research has been done on either non-humans or humans with memory difficulties. The relevant research with humans tends to be of low quality with few participants and poor controls. And lastly, for each purported nootropic, there are typically a few positive findings on which the vendors focus. However, these findings are often difficult to replicate or represent very small effect sizes. In short, no purported nootropic has been convincingly shown to have memory-enhancing effects. There may be enough positive evidence to warrant continued investigation of some potential nootropics, but there is not nearly enough to justify the various claims that are made in advertisements for these substances. This chapter, this chapter began with the case of HM and it ends with the case of RM. So RM is a biopsychologist and you will learn how his vocation played an important role in one of his symptoms. RM fell on his head while skiing. When he regained consciousness, he was suffering from both retrograde and anterograde amnesia. For several hours, he could recall few of the events of his previous life. He could not remember if he was married, where he lived, or where he worked. He had lost most of the episodic memory. Also, many of the things that happened on him in the hours after his accident were forgotten as soon as his attention was diverted from them. For example, in the car on his way to the hospital, RM chatted with the person sitting next to him, of a friend with whom he had skied all day. But each time his attention was drawn elsewhere, for example, by the mountain scenery, he completely forgot this person and reintroduced himself. This was a classic case of post-traumatic amnesia. Like HMRM was trapped in the present with only a cloudy past and seemingly no future. The irony of the situation was that during those few hours when RM could recall few of the events of his own life, his thoughts repeatedly drifted to one semantic memory, his memory of a person he remembered learning about somewhere in his muddled past. Through the haze he remembered HM, his fellow prisoner of the present, and wondered if the same fate lay in the store for him. RM recovered fully and looks back on what he can recall of his experience with relief and a feeling of empathy for HM. Unlike HM, RM received a repri- a re- Unlike HM, RM received a reprieve, but his experience left him with a better appreciation for the situation of those like HM who serve a life sentence. Brain teasers time. Test yourself. Blank are a population of neurons that are activated by learning, having during cellular changes as a consequence of learning, and whose reactivation by a part of the original stimuli delivered during learning results in memory recall. The correct answer is engram cells. Blank is the cortex of the inferior temporal cortex. The correct answer is inferotemporal cortex. The blank is thought to play a special role in memory for the emotional significance of experiences. The correct answer is amygdala. A phenomenon where there is a facilitation of synaptic transmission following high-frequency electrical stimulation applied to presynaptic neurons. The correct answer is long-term potentiation, or LTP. And lastly, blank are substances that are thought to improve memory. The correct answer is nootropics or smart drugs. So, so as we end this chapter, let us revisit the four themes of this book: the neuroplasticity, clinical implication, evolutionary perspective, and the thinking creatively. So, the biopsychology of memory is a neuroplastic phenomenon. It focuses on the changes in neural function that store experiences. Thus, the neuroplasticity theme was implicit throughout the chapter. Because the study of the neural mechanisms of memory is based largely on the study of humans with amnesia, the clinical implications theme played a significant role. The study of memory disorders has so far been a one-way street. We have learned much about memory and its neural mechanisms from studying amnesic patients, but we have not yet learned enough to treat their memory problems. Animal models have also played a major role in the study of memory disorders. Only so much progress can be made studying human clinical cases. Questions of causation and questions of causation must be addressed with animal models. The study of medial temporal lobe amnesia illustrates the comparative approach at its best. Finally, the Thinking Creatively tab marked 3 points in the chapter where we were encouraged to think in unconventional ways. We were encouraged to consider why it proved so difficult to develop an animal model of medial temporal lobe amnesia, why cases of cerebral ischemia do not provide strong evidence for involvement of the hippocampus in object recognition memory, and the gulf between the actual evidence for smart drugs and the advertised claims.